Alright, good afternoon everyone. Welcome back to class. I hope you all had a good weekend. Yes? Say it again?
Oh, I had a 4-day weekend. It was great.
I know, it was great, yeah. But it's good to be back. And so, how many of you like to watch TV series which are super long, like 50, 60 episodes? Yes? Alright. What is the most exciting part of such long stories? The end, right? Exactly, okay, perfect. The end is really exciting partially because the way there are different players in the story, they may be in different geographical locations, how it all ties in together. And I think a really good storyteller is able to make everything tie together with no loose ends, right? I'm not that kind of storyteller. But when I think about human physiology and how the body works, I think it's kind of like a saga, the way the various players will eventually tie in together. Right? So that's the first thing point I wanted to make about that. Second point is, whenever you start a new episode, they say that previously on blah blah blah, they give you a very short recap of what happened in the previous few episodes, right? So that's what I hope to do for the next 30 seconds to 1 minute, so that you can get the continuation of what's happening. Now, if you are one of those who likes to binge watch, right, then you can say skip intro and you can move on to the next one. And I hope none of you are going to binge watch these lectures. That will be a real punishment, right? So I'll give you the intro now, and yeah, please make sure that you stay with these lectures because it will be a lot at the end. So if you were to tell me what is the one thing we've talked about, right, in one word, like we talked about a few things these last 2 weeks, but if you could describe it in one 4-letter word which starts with the letter C, cell, right? We're talking about what happens in a single cell. And there's only one small piece of information missing in what I hope to tell you, and that is this: cytoskeletal and motor proteins. Right? So that is the last bit I need to cover, but let's see how this last bit fits into the functioning of a cell. So when you look at the cell, the major component in it is made up of what? Water. Thank you. Yeah. Thank you. Yes, water is the main component. And that brought us to the discussion of hydrophobic and hydrophilic interactions. And we also looked at this cell membrane, which has a hydrophobic and a hydrophilic region. Another thing in the cell membrane were a number of transmembrane proteins. And the question was, where do these proteins come from? And the answer was explained through the central dogma. So in the central dogma, the first step is DNA to RNA that takes place inside the nucleus. RNA to proteins takes place mainly in the cytoplasm, but it also takes place in organelles like the endoplasmic reticulum, where there is further modification of the proteins. And then we see the endoplasmic reticulum also not only helps make proteins but also degrades proteins which are abnormal. And that's how it maintains proteostasis or protein homeostasis. And from there, the proteins are moved to the Golgi apparatus, where they are given their destinies, right, or their destination through what type of modification takes place to it. So it could have 3 types of destinations. Number 1 is the lysosome, where it's degraded, or number 2, it could go and be a transmembrane protein. And number 3, it could be sent out of the cell. Now this destiny 2 and 3 requires exocytosis, right? Whether you get a transmembrane protein or sending the proteins out of the cell requires exocytosis. And we talked about the snare hypothesis to see how the vesicle can actually fuse with the cell membrane. We also talked about how substances enter the cell by talking about endocytosis, and we see that there are different types of proteins involved in carrier-mediated endocytosis. Right? And the last bit of information missing is, how do these vesicles get from one part of the cell to the other? And the answer to that question is right here on this slide. We have these cytoskeletal proteins on which there are motor proteins which can walk carrying the vesicles. Right? So this is the last piece of what happens inside the cell, which we hope to cover in today's lecture. So with that, let's look at a quick overview of what we'll be discussing. So we see that proteins are transported along the cytoskeleton, and there are three types of proteins we'll discuss, and only two among them will play a role in transport of vesicles. And the way these vesicles are transported is through motor proteins, and we look at three of them. One is kinesin, shown here, dynein, and myosin, shown right here. I told you that among these three types of cytoskeleton, only two play a role in transport. So we'll see what are some of the other functions of these cytoskeletons as well, right? Cytoskeletal proteins. So with that being said, this was the slide where we finished on Friday. So we see that in order to transport vesicles within the cell, if you consider how cargo is transported on land, it needs two things in our society. You need roads, and you also need vehicles on which those roads can go, on vehicles on which the cargo can be carried and it can travel on the roads. Right? So if you keep that analogy in mind, the cytoskeleton proteins will be the roads, and there are three of them, and then you have the vehicles, which are motor proteins. So keep in mind that only the microtubules and the actin filaments will play a role in the transport of vesicles. And you'll see that this kinesin and dynein, the first two, will transport vesicles on microtubules, whereas the third one, myosin, will transport vesicles on actin. So let's take a closer look at each of these. Now, the question is, why do we call one of the cytoskeletal proteins as intermediate filaments? And if you notice here, the answer is mainly because of the size difference. So intermediate filaments are around 8 to 12 nanometers in diameter, whereas the smallest cytoskeletal proteins we consider are actin subunits. That's around 7 nanometers, and the largest are microtubules, around 25 nanometers. So we see that the intermediate filaments have an intermediate size compared to actin and the microtubules. What we have in the above panels are also fluorescent dyed microscopic images. And notice that these intermediate filaments, they are starting at the center and go all the way to the periphery of the cell. This hole you see right in the middle, that is where the nucleus would be. If you look at the other two panels, you see the nucleus is it's labeled in blue, but you see that the intermediate filaments are found all the way from the center out to the periphery. And the intermediate filaments have only a structural role, and they will help in supporting the structure of the cell. Now, if you look at these other two panels, notice that the green color labeling is indicating microtubules. So it's found starting at the center of the cell, and it can extend all the way towards the end also. Right? Whereas the actin filaments, it's found more only towards the periphery of the cell. And these microtubules and actin filaments both play a role in structural support and in transport. And we see that because these microtubules are more extensive and long, all the way from the center to the periphery of the cell, it makes sense to say that it is involved in long distance transport. Whereas the actin filaments, they will be involved in short distance transport at the periphery of the cell. Alright, if we are all clear up to that point, we can move on to see a few more details about each of these cytoskeletal proteins. So notice here that here is a filament of a monomeric filament of what you have these intermediate filaments made of. And one of the proteins found in these filaments is keratin. And notice here that monomer has a N terminus, and there is a C terminus. The N terminus indicates the free amino group, whereas the C terminus is indicating the free carboxylic group in the amino acid chain. So two monomers can come together to form a dimer. And how would you categorize that dimer? Would that be a primary, secondary, tertiary, or quaternary structure? If you have two dimers coming together. Yes, quaternary structure, right? Because a quaternary structure is made up of multiple subunits coming together. And these dimers can form a tetramer, and that can eventually link together, and you have these intermediate filaments. So as I mentioned, they play a role in structural support because they are rope-like. When you have multiple filaments coming together, and that adds a lot of strength while still keeping the filaments quite flexible. And you will find these intermediate filaments made of keratin, and it's found in the hair, nails, and it's also found within the cells, offering structural support. And when we talk about cell junctions later in today's lecture, and also when we come back tomorrow, we'll see how these intermediate filaments actually play a pretty important role in it. But let's move on to those cytoskeletal proteins which are helping in transport. Did you have a question back there? No questions. Okay. So when you look at microtubules, they are made up of these individual subunits called a tubulin. Right? And there are two types of tubulins. You have an alpha and a beta subunit, and both will combine to form a dimer. And the diameter of this microtubule is around 24 nanometers, so it's like a hollow tube. And you notice that there are 13 such tubulin subunits which come together to form this hollow cylinder. Keep in mind that this microtubule is going to have an orientation where one end is known as the plus end and the other end is called the minus end. This has nothing to do with electric charge, but the plus end is referring to the part of the microtubule which is towards the cell surface, and it's facing the cell membrane, whereas the minus end is towards the nucleus or the center of the cell. And that is all the detail I will give you about the plus and minus ends. If you take a more advanced course in cell biology, you will understand why those plus and minus ends have relevance to the assembly of the microtubule complex. Right? But for now, just know that plus and minus ends, they are referring to the directionality in the cell. So there are a few functions of these microtubules. We'll talk about the long distance transport and how that happens using two types of motor proteins. And then we'll also talk about how it plays a role in cell division when we consider the reproductive system. So in the reproductive system, we'll talk about mitosis and how you have two identical cells formed by cell division. And in that, the microtubules will play a role. Moving on to the last type of cytoskeletal protein, we consider filamentous actin. That's what is referring to as F-actin. And this filamentous actin is made up of monomeric subunits called G-actin. So G stands for globular actin. And all of these globular subunits, they're going to arrange into a double-stranded filament. So notice how it's wrapping around each other. You have one strand shown in light blue, the second strand shown in green. So you have two strands bound around each other. Now, this has a number of functions. We see that it will help in maintaining the structure of the cell, and I'll give you specific examples of this when we come back tomorrow and talk about a type of cell junction. It also plays a role in short distance transport, and we spend quite a bit of time talking about how these actin filaments will interact with myosin molecules to generate force and causes muscle contraction. And this is found not only in skeletal muscle, it's also in cardiac muscle and in smooth muscles as well. And finally, actin can also play a role in cell division. So now that we've talked about the different types of cytoskeletal proteins, we can move on to talk about what are the motor proteins which will move on these cytoskeletal proteins. Keep in mind that all of these motor proteins are ATPases, and that means these are enzymes which can break down ATP into ADP and inorganic phosphate. And the energy which is within those phosphate bonds of ATP will be used to power the motors to move them along the cytoskeleton. So we know how the ATP was made when we talked about metabolism. It's through breakdown of carbohydrates and fats, and now we see how the ATP is actually being used in the cell. So there are three types of motor proteins we'll discuss. We have kinesin shown here, and then followed by dynein below that, and finally myosin. So now let's see how this kinesin can actually help transport vesicles. So just watch this short video clip here. This is a large vesicle, and it's bound to a kinesin molecule at one end, and the other end of the kinesin molecule is bound to this microtubule. And notice that these vesicles have a number of transmembrane proteins on them, and there are two foot proteins which are alternatively binding to the microtubule. The main detail I want you to take home is that kinesin will walk on microtubules towards the plus end, and this means it's moving from the center of the cell towards the cell membrane. An additional detail which I will not hold you responsible for, but it's for your curiosity's sake, is that it takes around 100 steps per second, and it moves centimeters per day. So the question is, how does this work? Alright, and so let's look at this cartoon here. Notice that there are 2 feet shown here for the kinesin molecule. You have 1 feet in blue, and the other foot is in purple. And the question I want you to ask yourself is, I see that the purple foot is bound to the microtubule. What will cause the other foot to take the next step forward and bind to the microtubule? So the question you want to ask yourself is, what can happen to make this blue foot take the next step forward and bind to the microtubule? So notice that there's ATP shown here as a yellow triangle, and you have ADP, which is shown here in orange, which is the byproduct when ATP is hydrolyzed to ADP, and the other product is the inorganic phosphate shown here in green. So in order for this blue foot to move forward, the purple foot has to be bound by ATP. So once ATP binds to the purple foot, the blue foot, which is hanging there, is going to swing forward and then bind to the microtubule. Notice that this blue foot had ADP attached to it, and the reason it had ADP attached to it was because it had previously hydrolyzed ATP to ADP and inorganic phosphate. And when this blue foot binds the microtubule, notice that it is going to lose the ADP molecule. And now this blue foot will be the same position as the purple foot in the previous step. There's nothing bound to it, but the purple foot is bound to the microtubule. Now, the ATP which was bound to the purple foot will also be hydrolyzed, and it's going to release the inorganic phosphate, and once it has the ADP attached to it, it's also going to release its hold on the microtubule. And so the question is, what makes this purple foot move forward and bind to the microtubule? And the answer is, when you have ATP binding to the blue foot, the purple foot can take the next step forward. Right? And this step will repeat over and over again. And the first time I heard this, it didn't make sense. Right? So I would highly recommend that you walk through each of these steps to know what causes the foot to move forward. So if one foot is hanging at the back, ATP should bind to the other foot which is attached to the microtubule to make the foot move forward. Yes?
What's being discussed? What are we going to do to produce the
So your question is, where does this phosphate come from, right? So this here is ATP, right? Shown here as the yellow triangle. It will be hydrolyzed to ADP and inorganic phosphate. So the ADP is still remaining bound to it, whereas the phosphate was released in the previous step.
So it's just the third phosphate.
The third phosphate is released. Yeah. Any other questions at all? Walk through these steps, folks. Yes.
It seems kind of like inefficient. Like it sounds like there's a lot of ATP involved, in just like one small thing.
Yeah, it is expensive to keep the cell alive and working. A lot of ATP is used. And even a side note, not even all of the energy in the phosphate bond is released. Only around 40% is used to do work. The rest is lost as heat. So, yeah. Anyway, so that's how the kinesin molecule moves forward on the microtubule, and it's moving in the towards the positive, the plus end of the microtubule. Dynein is also going to move on the microtubule, but you'll notice that it's moving towards the minus end, and it's slightly faster than kinesin. It's around 88 millimeters per day. And here's a short clip of how dynein walks. Right? So it's also moving on microtubule, and it's walking like a drunken soldier or a sailor, and it's using ATP for each of those steps. All right. So finally, moving on to myosin, notice that myosin will interact with filamentous actin, and you have different types of myosin proteins. So myosin 5 will move towards the plus end, it's around 20 millimeters per day, and the myosin 6 will move towards the minus end in the opposite direction, around 5 millimeters per day. So these are just additional details on the speeds of these motor proteins. But keep in mind that the myosin, it has a much slower speed because it is traveling a shorter distance, unlike the kinesin and dynein, which is traveling a much longer distance. So with that, I'd like to summarize what we've discussed till now. We see that cytoskeletal proteins, they not only maintain the structure of the cell, but some of them can be helpful in transport of vesicles. And the intermediate filaments are mainly for the support of the cell, and it's made up of keratin, and it can also play a role in the adhesion of cells to one another. And as far as transport is concerned, microtubules can help in long distance transport by permitting both kinesin and dynein to walk on it in the towards the plus end and the minus end respectively. And finally, the F-actin can help with short distance transport by the action of different types of myosin proteins. Right. So with that, we can do a couple of key questions, which I hope we can answer. So what are the three types of cytoskeletal proteins and how are they formed? Right. So I'm sure you can answer what those three are. You have actin filament, microtubules, and intermediate filaments, right? And then how they are formed, you could just say what are the individual monomer subunits and how they are connected. So how do you think microtubules are formed? What are the individual subunits? Alpha and beta tubulin, that's right. And then what about the actin filament? What are the usual subunits? Globular actin, G-actin. And for the intermediate filaments, you have those long filamentous fibers which can form dimers and then tetramers, and keratin is one of the constituents of those intermediate filaments. What are the different types of cytoskeletons? We already talked about it, right? Intermediate filament, actin filament, and microtubules. And the functions, how do they differ in function? So for structure, it's intermediate filaments, but for movement of vesicles, it's microtubules and actin filaments, right? There are some additional functions I mentioned, such as muscle contraction, or like cell division. So keep those additional points in mind as well, in your mind as well. And how do motor proteins transport vesicles on the cytoskeleton? So yes.
They walk using like ATP and the microtubules.
So they walk using ATP and they walk on the cytoskeletal proteins. So what walks on microtubules? Myosin doesn't walk on microtubules. Kinesin and dynein, yeah, both walk on microtubules. And what walks on actin? Myosin, very good. Okay, with that, we can do a few questions from Friday's lecture, alright? So what are the functions of smooth and rough endoplasmic reticulum? What is smooth endoplasmic reticulum used for? One clue is, what does the liver do? You have a lot of smooth ER in the liver. One of the functions of the liver. Detoxification, so the smooth ER plays a role in detoxification. It also acts as a storage for calcium, and which is a secondary messenger, and it also helps in synthesis of what? Steroid hormones. Yes, that's good. And what about rough ER? It helps in proteostasis. Very good. So rough ER will help not only synthesis of proteins but also degradation of proteins. And what is the function of the Golgi apparatus in respect to protein modification? Yes.
Golgi apparatuses are essentially the post office of the cells. They will contain proteins
Mhm.
and make them for their clinical custody in the cell. And
Yep.
it's in the 23 locations.
Great. So I don't know if you others heard, but the Golgi apparatus will act like the post office, which collects the mail, sorts it, and then sends it to various destinations. And I kind of like what you said. It determines the destiny of those different proteins. So there can be 3 destinations. Destination 1 is where? The lysosome to degrade it. Destination 2 would be the cell membrane, to be a transmembrane protein, and destination 3 would be get rid of the protein, release it outside the cell. It could be a hormone, right, where it can have another destination where it needs to go bind to. And then, how does the endoplasmic reticulum maintain proteostasis? We already talked about its role in protein folding, but how does it help to keep the abnormally protein level down? How many ways does it use? How many ways? The ER does it maintain proteostasis? 10, 2, 3. What number would you choose? 5? Wait, 3, yes. There are 3 ways in which it maintains the levels of abnormally folded proteins. What is the first way? Starts with E. First step is ERAD, right? And what happens in ERAD? You remember? There'll be the ubiquitin tags added, and then those proteins are sent to the proteasome. Then you also have the second one, which is the UPR, which stands for. You look at your notes too. Unfolded blank response. Very good. Unfolded protein response, where you have either an increase in chaperone synthesis and a decrease in the abnormally proteins being synthesized. And what is the last step? The cell feels it can't keep up with the misfolded proteins. Autophagy, very good. The cell commits suicide. And how are proteins transported via the cytoskeleton? And the answer is motor proteins. We talked about it in great detail for the last half an hour. And what are the 3 types of protein coats used for receptor-mediated endocytosis, and when is each one used? I'll give you a clue. Sounds another word for police. Yes, cop. One,
Oh,
two,
cop one,
cop 2, and cop 3, right? So I hope you know when cop 1 is used, when cop 2 is used, and when clathrin is used as well, right? And what is the snare complex? How many proteins did we talk about in the snare complex?
Probably 5, right?
4 actually. 2 on the vesicles and 2 on the target membrane. And we talked about synaptotagmin on the vesicle, synaptobrevin or VAMP on the vesicle again, and there are 2 on the targets. One is SNAP25 and syntaxin, right? And when calcium binds to one of those proteins, it causes that whole complex to have the helices wind together, and that's how the vesicles can fuse with the membrane, and then exocytosis can take place, right? So with that, let's try to answer some few key questions. We'll answer a few polar beaver questions. So the presence of a single peptide in a protein sequence indicates that it will least likely be processed in the blank. Is it rough ER, Golgi apparatus, or cytoplasm? Or is it all of the above are possible? Okay, we have over 100 responses. Okay, the right answer is the cytoplasm, right? Because if it's got this signal peptide on it, it will end up in the ER, and from the ER it can go to the Golgi apparatus. So if it's least likely to be processed in the cytoplasm. What is the last resort for ER stress due to unfolded proteins?
It's locked.
Is it locked still? Okay, so most of you said autophagy, and that is the right answer, right? The very first step would be ERAD, as we talked about. The second step would be UPR. The third step would be ERAD, and proteosomes are involved under ERAD, so that would be under the first step. Moving on to the next question. Which of the following proteins are most directly affected by changes in calcium concentration? Is it VAMP or synaptobrevin, or syntaxin, or synaptotagmin, or SNAP25? So I really need you to know what the various players are in the SNAP complex and what their functions are. So which of the following do you think will be affected by calcium concentrations? So synaptotagmin is the correct answer. So that is the protein which binds calcium, and when that bound by calcium, it causes the other 3 proteins to cause them to wind together, and then the vesicles can fuse for exocytosis to take place. Alright? Okay. And then mutations in which of the following will affect vesicle transport from the Golgi apparatus to the endoplasmic reticulum? So I told you there are different types of proteins involved in vesicle transport, and this is receptor-mediated endocytosis. So which do you think plays a role in the transport of vesicles from Golgi apparatus to the ER? So most of you said COP1, and that is correct. So Golgi to ER is COP1, ER to Golgi is COP2, and from the Golgi to the plasma membrane and back is COP2. Right. So I hope that's clear, folks. Looks like most of you got these answers correct. If anything isn't clear, please go back to the slides and see if you can find the answer. Right? And if it's still not clear, talk to the TAs, and if it's not clear, then talk to me. All right. So now we move on to cell junctions. So up till this point, we've been talking about what happens within a single cell, but now we'll see how these cells are held together, and that is how they can work with each other, and that's how we have this full body, right, where cells are working together. So let's begin with an overview of what I hope to cover in today's lecture. So the question is, how are cells connected to each other and the extracellular matrix? And the answer is in three ways. Right? The first one is through gap junctions. And gap junctions, they will basically connect the cytoplasm of one cell to the other through these junctions, through which there can be molecules and ions passing from one cell's cytoplasm to the other. The second type of junction is called tight junctions, and this will form a seal between adjacent cells. So it's like a seal between the lateral surfaces of the cells, so nothing can get in between them. And thirdly, we have these anchoring junctions. Their main role is to provide mechanical support by connecting cells together. And it is in this third types of cells where type of junction we'll talk about the role of the cytoskeleton. So we'll see how the intermediate filaments and actin can play a role in anchoring junctions. So in today's lecture, for the remaining time, we'll just talk about gap and tight junctions. We'll come back to anchoring junctions in tomorrow's lecture. So gap junctions, they're going to mediate cell-to-cell communication because of these intracellular channels which are connecting the adjacent cells. And the question is, how are these channels formed? You have these individual subunits called connexins, and 6 such connexins will come together to form a hexameric protein complex. And notice that in one cell you have these 6 subunits. It's going to connect with the adjacent cells 6 subunits, and that's how you have this intracellular channel being formed. Now when these 2 intracellular channels are being formed, keep in mind that both of them are transmembrane proteins, and when they come together, there will be a small gap in between the 2 membranes, and that is the reason why it's called gap junctions. Right? So there are these gaps formed between the membranes because of the way in which those hexameric subunits come together. So this is just an electron micrograph showing you what this gap looks like. It looks like a black line, right? But this is just for your curiosity. I'll just put a small insert of this picture for your reference. So we see that because of these gap junctions, the cytoplasms of 2 cells are connected, and as a result, ions can flow across and metabolites can flow across. Metabolites are like carbohydrates or amino acids, and so we see that these cells are coupled both electrically and metabolically because of these gap junctions. And it was initially thought that only excitable cells like nerve cells and muscle cells will have gap junctions, but it's been found in other parts of the body as well. For example, we'll talk about even how the osteocytes, which are found in your bones, even they communicate with one another through gap junctions. So we will see how these gap junctions can play an important role in the functioning of different types of cells in the body. So when you think about having gap junctions in different tissues, keep in mind that there are tissue-specific isoforms which are expressed. So what do we mean by the term isoform? So isoform is a protein which is found in different types of tissues, and in various tissues there will be slight modifications which will help that tissue-specific function. And let me give you an example. Right? We have cardiac muscle and skeletal muscles. In both of these muscles you have actin-myosin interaction, but the myosin protein in the cardiac muscle is quite different than the myosin in the skeletal muscle. If you were to switch the myosin isoforms, the function of that organ will be affected. Right? So this is an example of how there are different types of isoforms in different tissues. So when you look at these connexons, there are different isoforms which form heteromeric subunits. So you have maybe a couple of isoforms coming together with different other isoforms. And by having these different isoforms, you have two things which are accomplished. You have variability in the permeability of these gap junctions, and secondly, you also have a regulation of these gap junctions. So what do we mean by regulation? So when you think about temperature being regulated in your room, right, you can either increase or decrease the temperature. That's a way of regulation. You can adjust the speed of the car you're driving, or even turning off of the faucet is a way of regulation. So these gap junctions, they are not always open. They can be open or closed. And what controls the opening or closing is the surrounding signals. So it could be a change in pH, it could be a change in ion concentration. So those things in the environment can cause a regulation of these gap junctions opening or closing. Another way of regulation is also where you have an expression of these connexons to be produced. And keep in mind that for these connexons to be incorporated into the cell membrane, it has to be done through exocytosis. So I hope you can see how the concepts which we have been discussing, right, these last 2 weeks, how they're all incorporating and being integrated as we consider how the various cells function, and also how even the gap junctions are functioning. Did you all have a chance to write that down? Inserted by exocytosis? Alright, in that case, we can move on to the second type of cell junction, and that is tight junctions. And so this is just an electron macrograph showing you gap junction in this panel here. And adjacent to that, you have a tight junction as well. At this magnification, you may be wondering, okay, nothing seems very different. But then if you take a closer look at the gap junction, you see this small dark line. That is actually a 2 to 3 nanometer gap between the two plasma membranes. So moving on to tight junctions, we see that they are holding adjacent cells together to form a near leak-proof seal. And I need to qualify why I use the word near leak-proof, because tight junctions are not, they can be selectively permeable depending on what types of proteins are expressed. And that's why I say it's going to have a near leak-proof seal. Notice also that these seals are formed between the lateral sides of these cells. And I want to give you an example of the cells found in your digestive tract. Right? So here's our digestive tract. This is the lumen in your gut, and this is showing the basal part of these cells. And notice that these tight junctions, they are found towards the apex of the cells, or towards the apical surface. And whenever you consider epithelial cells, the part of the cell which is exposed to the outside world, that is the apical surface. For example, we have epithelial cells on our skin. The apical surface is exposed to the outside world. The basal surface is connected to the underlying layers. Similarly, in your digestive tract, whatever you eat is going through your GI tract. That is the external world, and the apical surface will be facing that. So when you have eaten carbohydrates and you have glucose being found in your digestive tract, it just can't enter in between these cells. It has to enter through specific transport proteins. And the way you can think about tight junctions is like having a fence around your house. The presence of the fence means that there's only one way you can enter the house. It's through the gate. If you didn't have the fence, you can enter the area any way you want. But because of the tight junctions, the glucose can enter in only through specific transport proteins. And once it enters into those cells and goes into the bloodstream, it can't just exit back through the gap between the cells because of the tight junctions. And keep in mind that there are different types of isoforms in these proteins which make up tight junctions. And so permeability to the ions and other molecules can really vary based on what tissue you're taking into consideration. For example, in the small intestine epithelial, it is many-fold permeable to sodium when you compare to the urinary bladder. That's just an example of how permeability can vary. So now let's see what are the proteins which are found in the tight junctions. So notice here that there are some proteins which are found in between the cells. These are transmembrane proteins, whereas those which are inside the cell, that's called a plaque protein. So you have these claudins shown here in light blue. Below that you have occludins, and then you have these junctional adhesion molecules. So all of them are found outside the cell. They're providing external support. And whereas inside the cells you have these zona occludins. They are going to form the plaque proteins, and they are connected to the actin filaments, which are part of the cytoskeleton. So claudins are the main transmembrane proteins, and I will hold you responsible only for claudins when we talk about tight junctions. Occludins is another type of protein which is also found in the tight junctions, and zona occludins will form the scaffolding or support inside the cell. So let's look at the few isoforms of claudins. Right. Notice that this example of a claudin isoform, it is forming a pore, and so it can be selectively permeable. Whereas if you look at the panel below, notice that the claudin isoform does not form a pore, so nothing can get through it. So we have around 20 different isoforms in humans, and we see that different isoforms are expressed in different parts of the body. And so if anything goes wrong with these claudin proteins, it can lead to different kinds of pathologies. Right? And so if you look at this table here, it's really small because I don't expect you to read it. If you're really curious, click on this link here. But we see that there are two kinds of pathologies linked with claudin dysfunction. And the first one is cancers, and the second is inflammatory bowel disease. And we see that when the barrier, which should be formed by the tongue junction, is affected by claudin dysfunction, it can lead to inflammation as bacteria comes in, or it can also cause tumorigenesis when you have these epidermal growth factors. So let me explain how this happens. So in this cartoon here, notice that the darkly colored cells are indicating those cells which have a dysfunction of claudin, and as a result you have bacteria entering and these epidermal growth factors. And as the bacteria comes in, it can trigger an inflammatory response by releasing cytokines and activating T cells. Now both of these things will make sense when we talk about the immune system, but keep in mind that its downstream effect is what I will hold you responsible for now. So it can affect the synthesis of claudin proteins, and that can further exacerbate the problem. Secondly, notice that also these growth factors can enter where they're not supposed to, and that can lead to the formation of tumors. And all of this can be prevented if you stop the claudin dysfunction. And so probiotics can help with mucosal healing and also the synthesis of appropriate claudin proteins, and that can prevent this from happening. So with that, I'll stop and we'll pick up with the third type of cell junctions, antigen junctions, when we come back tomorrow. All right? Thank you all for your time.